Welcome, everyone. Thanks for joining today's town hall of Arise. We are doing these types of town halls every quarter to inform you what we are doing around Arise, but also to get your feedback. So today really isn't just about us telling you we've done some amazing things in the past couple of months, but also to ask your feedback. So please, if we are done with our presentations and you have any kind of questions, any kind of feedback, or you just want to listen in and brainstorm with us on the next steps of Arise, feel free to join. We're going to make breakout rooms in Zoom, and you can choose your own breakout room that you want to join, but please do so. We are really looking forward to talk to all of you. So, Arise. Why Arise? Well, Arise is one of those bigger infrastructures that is evolving around the biodiversity crisis. Obviously, most of you will know how important biodiversity is for our lives, our planet, our everything, basically. And luckily, this topic is getting more and more attention. So capturing the loss of biodiversity, or even just biodiversity in general, is very critical for our future. And that's hard, because we actually don't know how many species we have. There's an estimate of 43,000 species in the Netherlands alone, but we've only been able to actually recognize a small portion of this. That's mostly also because many of these species are in these hidden realms, so in the soil or in places that are hard to identify. So we need different technology. But also for these species we do recognize, it's a very laborious and intense sampling technique usually necessary. So how can we actually scale up and make biodiversity more accessible for everyone? That starts with species identification. Of course, we know it's not just species identification. As soon as you know there is a species, there's a lot more questions to be answered and a lot more data to capture, but at least it's a starting point. So one of the key missions of Arise is also to do this. Increase our knowledge, increase the technology that we can use for species identification. Then, of course, it's facilitating research. So what do you do once you have this information, once you have this data? Can you mix data? Can you use different types of data sets and connect it so that you actually can answer bigger questions? And last but not least, trends in time. Biodiversity right now changed. How much? For which species, under what conditions? Only then can we actually start influencing also the outside world and improve our situation. So what do? We basically have three main pillars in Arise. It's high throughput sampling and sequencing from a single species to our environmental samples. We need to have standardized semi-automatic processes to do so. So really have this is how we're going to do it, and we're going to do it fast, and we're going to do it cheap. But you also need transparency, because a lot of people will be involved in capturing these kind of samples, handling the samples. We also need to know who's done it, done what time, so we can trace back if anything was wrong, or we can change the data and why did we change the data, so that we also really have a core data set that is valid and our reference to refer to. A lot of data will come in Arise, and data alone is also not good enough. So a second pillar within Arise is not just to have a trusted database or multiple data sets to use, but also to have services on top of this. What do you do with data? And we'd love to know from you, for instance, in research, what do you need from the data to do? Can we do real-time identification, which is a service on top of the knowledge that we have? Can we create a digital platform where you basically come in and shop for the data that you need, or do identification, or build your own AI models? All of these types of questions will be answered in Arise. Last but not least, like I said, monitoring in the field is extremely important, and we have our more traditional methods, but can we complement those with technology? There's drones out there, there's all sorts of cameras getting cheaper and cheaper. So luckily, we can now also test out different novel ways to do this, not just on land, but also in sea or air. So that's also going to be extremely interesting to connect, and not just the species, but then also the environmental data. So how is Arise organized? Well, this is one of the big questions in the past few months. The last town hall, we talked about the architectural design study. So we really went into the Arise proposal, pulled it apart, created all kinds of blueprints, so to say, of what Arise could look like. But it also needed that transition from, okay, who's now going to do it? So we've built a Arise team, and we've organized ourselves in 5 different teams. And these teams are multidisciplinary, but they're also multi-organization. So Arise is a consortium. It's Naturalis, it's UvA, it's University of Twente, it's the Westerdijk Institute, which also means, you know, we have to collaborate. So why not do that within the teams? So these are our 5 teams. And of course, behind us are the big 4 institutes with also a wealth of information and expertise, which we also tap into. So we're not starting from scratch. We're actually building on top of knowledge, people, data, and services. So, but who to talk to for each team? And they're gonna take over soon and tell you all about what's happening in their teams. Sampling, Hanko just joined us over summer. Sequencing is Kevin Beentjes. For the digital species identification, contact Jacob. For BioCloud questions, it's Chantal. And for the monitoring demonstration sites, it's Daniel Kisling with his super assistant, Rotem. And new members are joining Arise, not just existing members from the institutes, but also a lot of hires that we've done over the past few months, which is great. So we're really building up our expertise. We're building up our team with people that can actually build an infrastructure. So we're going from thinking about it, dreaming about it, to actually building it very soon. We've also changed our ways of working, or at least we are changing them, because there was no way of working. It's an academic realm, but we needed a bit more structure if you're actually going to build an infrastructure. So over summer, we've also had agile training and coaching. We've been scrumming and starting to scrum more and more, which has been daunting, sometimes a bit painful, but certainly a good method to structurize where do we want to start? What do we want to focus on? How do we align our work across these 5 teams and make sure that we actually deliver value to our end users and not just what we think is the best thing to build. And that's been a theme for the last few months, but will remain a theme for Arise forever. Same for the continuous feedback. So maybe some of you might remember this picture. It's an overall view of all the data flows of Arise. And that's exactly how we want to work, end to end. So from getting in to going all out. And this can be described in a user story. So within Arise, we've now picked a few example user stories that we want to start with. So that we can, for instance, if we take image recognition as an example, you need to collect this. We've chosen one of the cameras. This is not the only one, but we're starting with this one so that we can collect the data, but also understand how we need to store such data. Then, of course, really process the data and do the identification of species. And then expose the data to you, to the research world. What do you want to do with it? And then, hopefully, also explore the data, add other data, maybe visualization. Maybe you can download it into your own virtual space and explore further or model further on top, or create your AI models on top that can benefit the rest of the community through Arise. All outstanding questions, but starting with one of these user stories will help us build the core components so that later on, if other devices are equally connected, we already know how to build this, and it's just a smaller step to scale up to other opportunities. So this is on the one hand, and you see underneath that all teams are involved. Sampling, monitoring demo sites, the BioCloud team, sequencing and DSI, BioCloud again, and of course UX UI. And if we move to another user story that we've prioritized, more on the traditional sampling and sequencing side, you see the same thing. You also need to collect, store, ID, expose, and explore. And by setting up these lines of components, hopefully you can build up Arise iteratively and fast. That's it for the general update. I'm going to hand it over to Hanko. So Hanko, if you unmute yourself.
Yes, hi everyone. I'm Hanko. And if Elaine goes to the next slide, I have a little photo of myself, and yeah, that's me in the collection. I'm brand new to the whole team. I started 2 weeks ago, so and I am excited. And my background is in the collections, so I already have some involvement in Arise before by information and etc. And also in the sampling team, Maurice has started. That's the next photo you quickly saw. She will be the collection manager, and she will be a major player in the sampling team, which is really nice. So to go to a little recap, which is on the next slide. Our focus team sampling is actually the whole process from in the field all the way to the lab, photographing, sequencing, and eventually storing for like probably eternity. So that's shortly spoken our main goal. And it all starts with collecting, making sure that we can identify species. And it involves a lot of processes. So we have to test and improve a lot of tools. We have like probably at some point 50 or 100 people in the field collecting for us. So we have to streamline a lot of processes. And it will also requires a lot of feedback loops, because we have so many people to connect with and make sure that everything goes in the same way or in an efficient way. And eventually everything will be, well, not everything, we will photograph all the voucher specimens that will be used as a reference database, and everything will be stored for eternity. And these will provide a huge database that can be used for all sorts of tools and goals that we have during Arise. For example, we try to implement image recognition to family and also for identifications, and also integrate this with sequencing data. So there's a lot to do, a lot of goals, and this makes me exciting. It really is a nice project, and So for the next months, which is the next slide, we will start easy, because we have to set up a lot of work processes for the collection, but also for specimens collected in the field, and make sure that this can be streamlined. And we already received the first samples. And not everyone uses, for example, data spreadsheets. So all the data is in a different way. So we need to streamline this and a lot of those kind of processes. And that's one of our targets for the next months. And we'll also start a small pilot in the collection, which we call collection mining. Basically, what we do is search for every species of a specific group in the collection and find the specimens, see if we got Dutch specimens, and sample them, photograph them, and make sure that we can already start making this reference database based on what we already have, because we have already so much actually. So yeah, that's our focus on the next month, and I think this is well within 5 minutes.
Well, onto the sequencing. This is just a title slide, so we can skip this one. So our main goals within the sequencing team are to basically optimize the entire process from DNA extraction to clean sequence data in a nutshell. What we're working now is mostly to reduce the cost and to ramp up the high throughput so that we can process all these species together. And of course, in the background, we do want to uncover all the genetic diversity of Dutch biodiversity. So next slide. So what have we done? Well, of course, our team is growing, and it's growing quickly. We added some technicians, an IT specialist, and a bioinformatician in the last couple of months. And we are now orientating on all the different protocols to optimize all these steps in the DNA workflow. So we are basically doing a lot of pilots. I'll tell a little bit about some of them. Some of them we'll discuss in a later stage. So on the next slide, so one of the things we are looking into now is, of course, the cost efficiency. As you can see in this graph, doing a single sample is almost 10 euros from start to finish. We have to do 350,000 specimens, so that's going to be a ton of money. So one of the things we are optimizing, well, two of the things we are optimizing currently are those two big blocks. You see the red one and the green one. So on the next slide, we can see the first step is the extraction. We managed to narrow down the extraction to about 5% of the original cost by throwing away all the expensive kits and switching to a homemade BOM protocol. Unfortunately, BOM protocol might probably put us on the no-fly lists because we are all Googling this protocol every day. But it seems to be working, so this looks to be the way forward for us. The next step is, of course, the sequencing. Old school Sanger sequencing is a little out of date. So we want to transfer all our sequencing to next generation platforms. So one of them is the MinION nanopore sequencing, which roughly reduces the sequencing cost to about 10%. And as you can see on the next slide, oh, the next slide is gone. We already have some good results with nanopore sequencing, so that's great. However, we are still we still have some road to go because, of course, you can still see like now most of the costs involve plastics. So we want to get rid of all the plastics, but we need some equipment for that. And we're currently looking into seeing if there's any possibility to buy some of this equipment. Also because, yeah, we kind of want to reduce our plastic waste as a biodiversity research institute. So other things we are doing in the next couple of months is adding even more people to the team. Like I said, we need to do 350,000 specimens. 1 or 2 technicians and alone is not enough. So we're adding a few more. And we're developing our protocols for the workflows. And of course, streamlining all the bioinformatics pipeline as well, because sequences are nice, but we want clean reference databases with barcodes attached to species names. And I think that's it for me.
Okay, good afternoon, everyone. My name is Jacob Kanninga, and I'm team lead for the Digital Species Identification team, together with a number of other people, which I put on the next slide. So it's a mix of Naturalis University people. So we can move on to the next slide. I guess most of you know, but I'll quickly go over a few use cases. So to give you an idea what we intend to do with DSI. Think about stationary sensors, for example, wildlife cameras to detect wildlife, the DFSS insect camera, which we are currently focusing on, but also bird sound loggers to listen to bird sound and identify birds that way. Same thing for bats, but also underwater sensors for marine or freshwater environments. You can also think about mobile sensors. That's the next slide. So think about cameras on drones or aquatic drones underwater, which are already being used to identify and count flowers, animals like sea lions, as you see here in the picture, but also zebras, rhinos, elephants, etc. Next slide, please. To give you an idea what we are designing and why is because we're facing many great challenges. It's in the word biodiversity. So indeed, the next slide, all of the things we have to accommodate are also diverse. There are many different species. Each species has many appearances. They live in different environments. There's different ways to record them. So different digital media types. And of course, there are many types of users who want to use DSI aspects in a different way. And we want to build something that accommodates all of these things and is scalable in the long run. So next slide. To give you a quick update, what we have done in the last months, we are also building a team, and very recently we hired our first software developer. So welcome, Patrick. And we are currently doing interviews for a second developer, both at UT, a second and a third developer, both at the University of Twente and Naturalis. So then we actually have people who can work on building the things full time. So far, what we have mostly been doing is designing the infrastructure. With the focus on 2 demonstrators, one of which is the Diopsis camera. I put Rotem her nice picture on this slide. And at the same time, we want to use the same infrastructure to deploy bird sound. And we're doing this so that we can make sure that what we are building is actually generic to different media types and different projects. And in order to do that, we have been designing an AI algorithm repository, and we are working on a deployment module. And to give you an idea what that means, I included a few slides. So these are actually in the end of a DSI pipeline. So I highlighted them here in the bottom of this list of things that we still need to build. And in the following slides, so with the algorithm repository, we want to host various algorithms that can be used to cover different geographic regions, but also different media types, different taxa, etc. And we want to facilitate users to easily select different algorithms. So there can be existing algorithms to deploy and to update and to maintain, but also for an algorithm developer to start off with an existing algorithm and make it easy for them to update a model or improve a model. And at the same time, we also want to allow developers from all over the world to propose better algorithms to publish in our library. Next slide, please. So what we envision is something like this, where you can search for algorithms. So for example, best performing algorithms available, or algorithms that are sensitive to MOS, if that's what you are working on. And then in this algorithm repository, we have different algorithms. In this case, I just marked up a few versions for Diopsis, for example, the ISA camera. So you want to select an algorithm and then ultimately click a deploy button. So if you can click, Elaine, and this is, and then an algorithm can be deployed. And we are thinking about a Docker service that actually hosts our algorithms. Now this orange line seems very easy, but that's actually a lot of work. So that's what we will be focusing on. And then on the next slide, when an algorithm is deployed, a user can send images to our deployed algorithm, and then they will get back the identified species, which is the next slide. Yeah, including locations, biomass estimation, etc. And the same infrastructure, as I said before, also needs to be used for other use cases. And in this case, we will start off with deploying an existing bird sound classifier. Then we'll send sound files to our deployed algorithm and send back identified species. Okay, next slide. So we'll finish hiring software developers to extend the team and actually start building. So hopefully next town hall, we can show you already some first demos. And I think those were my slides.
Yeah, thanks, Jakob. I'm continuing. My name is Daniel Kissling. I'm the lead for the monitoring demonstration sites. Next slide, Elaine. So the main aims of that team or that part of ARISE is, on the one hand, to establish sites, pilot sites, where multiple sensors are deployed, mostly for the automated or semi-automated collection of images and the subsequent evidence of occurrence of species. We also want at those sites to demonstrate the innovation and the potential of innovation for new sensors or the deployment of sensors, and also develop the knowledge that we need for this kind of automated monitoring, mostly related to image, sound, and radar. But we also will collect eDNA data at those sites. And finally, we want to connect with those sites also to users, to scientific users, to the stakeholders, to monitoring stakeholders, and the public. I want to quickly go through some of the sites we've been worked on in the last couple of months. The first one is Artis, where we have a good connection. It's mostly focused on urban biodiversity. It's not the zoo animals that we're focusing on. We are interested in the urban wildlife, but also it's an excellent site for public outreach and to demonstrate and test various sensors in the close vicinity of our university. The second site that we have been working on and deploying camera and wildlife cameras and insect cameras are the Amsterdam Savanne Down. It's a dune ecosystem, a very typical ecosystem in the Netherlands. It has also some technical challenge. It's relatively remote. So for instance, the mobile network is not that stable. So 4G data transfer is quite a bit of a challenge. But it has also, from an ecological point of view, for instance, grazing exclosures that allow us to assess disturbances and how biodiversity reacts to this. The third site we have been working on is an agricultural field site in Flevoland, from the crop field margin to from a flower strip at the crop field margin to the center of a field. It's an agricultural site. It's quite interesting in terms of farmland biodiversity and how insects respond to in agricultural settings. And the fourth site that we have been working on in the last couple of weeks is Oostvaardersplassen. Many of you might know as a rewilding and restoration site. It has quite a few different types of ecosystem, wetlands, and aquatic ecosystems, but also grasslands. And it's very famous for the large herbivores and also the effects of grazing on biodiversity. So at all those sites, we have been closely working with a range of stakeholders. These are, for instance, Artis Zoo, Waternet, farmer associations, or the Staatsbosbeheer. At all those sites, we are starting or have already deployed various types of equipment, of sensors. Here are some examples. We have set up a number of wildlife camera traps that work with a 4G data transfer. So data are directly transferred via the mobile network. We have established a number of low-cost acoustic devices like these audio moths. And we have established a bird radar at Artis Zoo. It's a bird scanning MR1 radar that allows to track migratory birds and insects. And then we have also testing and establishing a number of insect cameras. Diopsis we have already heard. And we have also tried out other equipment such as this automated moth trap and another intelligent insect camera with a GPU computer on board. For all these equipment at each of those sites, we try to define and develop scientific use cases. Here are 4 examples. For instance, the deployment of the Diopsis camera on the top in terms of insect biodiversity in farmlands, where we try to connect to ongoing research in a farmland site. We have, for instance, with the wildlife camera traps connected to Patrick Janssen, who's doing a lot of this wildlife monitoring at all sorts of sites to test the occupancy, but also the seasonal and daily dynamics of wildlife. In terms of the acoustic sensors, we have, for instance, connected to an urban rat population dynamic project in Amsterdam, where the monitoring of the rat population size is of very interest to the gemeente in Amsterdam. And the radar is, for instance, connected to ongoing research in bird and insect migration at the University of Amsterdam. Just to give you a few images, what we have collected here on the top are some images from the wildlife cameras, and on top and below are some images from the Diopsis cameras. Just in the last couple of 2-3 months, we have already collected nearly 38,000 images from the wildlife camera traps, and nearly 370,000 images from these Diopsis cameras. So you can imagine that this is a large amount of images, and all these images will then flow to the DSI, to this digital species identification engine, and we are at the moment establishing these data streams that allow then the connection to the DSI. And then I think the final one is just to highlight a few new or next steps. So we are at the moment mostly busy with testing and streamlining these data flows. So it's not only collecting those data, but how do you go from a 4G data transfer, for instance, to a server, to a portal that allows you to identify the species. Some of the devices that we have still require to collect SD cards, so they don't have an automated data transfer. We also explore data storage options, for instance, with SURF. And we also look into how we can actually then access the images and work with handling those images. The second big point that we are wanting to work on in the next couple of months is the data integration among those different sensors. Here's just a few examples. So for instance, how can you combine information from wildlife camera traps with audio moth devices? How can you maybe combine bird radar information with cameras or sound? And how can we connect environmental information from sensors, for instance, meteorological sensors? And then a last part is expanding the monitoring demonstration sites. At the moment, at this current sites, we also establish additional sensors, but we also want to expand our sites and to go to other sites, for instance, in Twente and the Hoge Veluwe National Park. And finally, also to develop, to deploy other sensors. So we're looking at the moment into GPS tagging, Nestbox cameras, aquatic sensors, phenocams, weather station. So all these kind of things will come in the near future. Thank you very much.
All right, I think I'm the last team to present to you. So my name is Chantal Huibers. I'm based at Naturalis, and I'm the team lead for the BioCloud team, which is a pretty diverse team of software developers, system engineers, data scientists mostly. We're based across all 4 partners that are part of Arise. And the BioCloud team is really responsible for developing the underlying infrastructure for the entire Arise program. As you have just heard from the other team leads, there will be a lot of data already collected within the Arise program that we want to connect also to data outside Arise, as well as lots of computational services and data workflows that we need to build. So to try to put BioCloud in a bit of a nutshell, it will exist of a range of end user services. So we imagine that there will be a platform at some point, not right now yet, but that is kind of like a one-stop shop with an interactive front-end layer where users can access data and services, visualize data, but also link that to other trusted data repositories such as GBIF and BOLD and Catalogue of Life, the Dutch Species Register, etc. In this shop will also be the algorithm library or repository that Jakob was talking about, and kind of like a digital playground or a sandbox where users can bring their own data, run an algorithm on it, as well as developers of other algorithms who can deploy algorithms or play with existing algorithms. There will be an annotation workbench where people can annotate data and enrich the metadata of data, and a storage portal. Well, before we can really build a shop like this, we need a lot of underlying services or major building blocks, as I like to call them, that really set up the services for managing all this data, where it is stored. So not everything will be stored in the same place. There will be data stored in relatively active storage where you can easily access it for analysis. But there will also be data that needs to be stored in archival storage where we don't lose it, but we also don't really need access to it immediately. To organize all of that, we are developing a digital object infrastructure. This is really defining a data model and data objects, ensuring that all the data that is in Arise gets a persistent identifier, so we can find it, we can add metadata to it, and we can actually query it to define new data sets built on particular search queries. We also really would like to control who has access to what, so that if an outside user accesses the Arise platform, brings their own data, that it's clear who can actually access this data. So make it as open as possible, but also as closed as necessary, as we sometimes say. And clearly there's a lot of links between all these data flows, APIs to external system, as well as internally within Arise that we're building. So if you go to the next slide, Elaine, thanks. So some people have already mentioned the Diopsis insect cameras. This is a workflow that we understand relatively well from end to end, from the cameras in the field that are collecting the images all the way to a report that comes out of it at the end for the end user. This workflow is largely developed by people within Naturalis and related teams. So for us, this is a very good use case to try to automate within the Arise infrastructure. At first, keeping in mind that we need to build the infrastructure not just for images from insect cameras, but also from other cameras, from other types of sensors, as well as for the whole DNA sequencing workflow. But we have to start somewhere. And as I said, we understand the Diopsys workflow pretty well. Therefore, this is one of the main use cases that we are trying to automate at the moment. So at the moment, most of these building blocks are in place, but some of them really require a lot of manual work, moving data from one place to another. What we're really trying to do is ensuring that this is better managed and available to others as well. So what you see in this image is a bit of a very broad overview of, we have cameras in the field. They can send images either as a batch or as a stream to a server. Well, before we can actually store this data somewhere, we really want to have better control over who can access this data, what is in this data, making sure that through a good setup of the digital object infrastructure, we can actually identify these objects and these images and make sure that when they are used in a particular algorithm, we can add metadata to that and put a data management system in place that understands where this data needs to be stored. And from this data storage, if you manage that well, you can then also more easily manage the flows to either a viewer service where people can view these images, a service where the DSI team can run the algorithms on them, or like a reporting system where you can query, give me all the images or the species identified in images from this region, and send that in a report. And at a lot of these steps, more metadata is added to images. So maybe they come in, as raw images with no clue what kind of species are on the image. But once it goes through an image analysis, this kind of data will be added. So the access control, the digital object infrastructure, and the data management system services will all maintain these structures and ensure that we manage this data well. Next slide. So for the next so we've been trying to design what this all needs to look like for the last few months, and continue to do this at least throughout the end of the year. So for the data management system, we are looking at using an open source software that is hosted by SURF. It's called iRODS. And without going into a lot of technical detail, this is a software package that's used by a lot of other universities within the Netherlands and internationally as well, where you can relatively easily, if you understand the system, can control where data is stored. So moving data between different storage systems, sharing it with different people, adding metadata, controlling metadata, and controlling who has access to it. SURF has already a lot of experience with hosting this. So we are working on a pilot with them to look at how we can implement this for Arise at the moment. And that will also allow us to more easily integrate this with other services that they can offer, such as storage solutions, as well as computational services. And another big chunk of work that we are working on is the whole digital object architecture. So making sure that all the data that we host in Arise is fair, giving all the data objects unique identifiers and appropriate metadata so that we can query it, create data sets for analysis, and share it with other people. For this, we are looking into using Cordra, which is also an open software package. And we're working closely with other projects such as Disco, which is also led by Naturalis, to look at how we can set this up. So a lot of building blocks to be built, but hopefully next time we can show you a little bit more what that looks in practice. Thanks, Elaine.
So just unmuting myself. Thanks a lot, everyone. And don't leave yet. A few spoilers. For the upcoming month, we also have 2 interactive design students that are joining our team. And the great thing about that is that they're all about, all of this technical stuff that's happening in Arise, but giving it an actual proper design. And by design, people think about, ooh, nice glass or a vase in their homes. But actually for data, it's extremely important that you are able to intuitively really interact with the data without getting lost in lots and lots of data flows or databases, really requesting from Arise what you want it to do for you. So we're happy that these students will join us, dig in, and see if they can actually give a face to Arise. So real prototypes where you can interact with the technicalities behind it. So it's time for your feedback. We've prepared some breakout rooms. So please don't leave yet, but pick one of the rooms when you're asked and join the discussion. And otherwise, see you soon for the next one in December. But first, breakout rooms, please. See a lot of you just, if you scroll down, you can pick any of the breakout rooms you want to join. So general, if you want to have more information just about Arise or ask generic questions. If you're really into sampling, join the sampling room. If you're all about sequencing and asking questions about this or the bioinformatics behind it, please join the sequencing room. The BioCloud room for Chantal's final talk. The digital species identification, so all about AI models. Join Jacob in the digital species identification room. And for the monitoring demo sites, just go to the field sites all the way in the bottom.
So Elaine, are the breakout rooms already opened? Because I can't see them.
Let me see. I can see them.
I can't.
That's interesting.
And I see others can't neither.
Ik ook niet.
Yeah.
Nee.
Okay, Bart, you're behind the... Thanks, Niels, for pointing that out. Because I see a lot of people still here.
If you move your cursor, a bar should pop up at the bottom of your screen with the mute and start video buttons. And then on the right of that bar, there's 3 dots that says more. If you click that, there should be an option called breakout rooms.
Yeah, normally that should pop up in the screen.
Then you get a long list of everyone who is in unassigned. But if you scroll down that list, there's actually 6 breakout rooms that you can join.
I'm not having the 3 dots.
Okay, let me stop sharing my screen. Maybe the message will actually pop up.
No dots for me as well.
I see participants.
Work in the web interface. Or in
the web interface is a bit more difficult indeed. Bart, maybe we'll just quickly create them again. Yeah, so we're going to close. We're just going to make some more. This is the first time we're trying this, so bear with us.
I'm not in the web interface and I don't have the option.
So, but Arjen, you do see the mute button and the stop video.
Yeah, and the participants and the chat and the share screen and the record and the reactions.